Gabriela Torrini. Today, I'm going to be talking about something that's not the center of attention when it comes to black hole searches. I'm characterizing the offset of intermediate mass black holes. Supermassive black holes are commonly found at the centers of large galaxies, including our very own Milky Way. They're upwards of 1 million times the mass of our sun. The gravitational pull of these dense objects is so strong that even light cannot escape them. However, astronomers aren't exactly sure how these black holes became supermassive. In smaller dwarf galaxies, the largest black holes would have an intermediate mass between 100 and 100,000 times the mass of the sun. To properly investigate the origins of supermassive black holes, it's critical to determine what role intermediate mass black holes play in dwarf galaxies. How common are they? Where do they reside? One model of galaxy evolution predicts that large galaxies formed via successive mergers between smaller dwarf galaxies. Since dwarf galaxies have a smaller central mass and they gravitationally interact during mergers, they could have a higher fraction of wandering black holes that are off-center. See the white arrows in the figure on the left for illustration. My goal is to test this hypothesis by measuring their offset from the center of the dwarf galaxy. But black holes are dark, so astronomers often have to detect them via the gas that swirls around them. One identification method uses the unique chemical fingerprint or spectrum of this gas. But traditional spectroscopic instruments only detect a small slice of the sky through a slit, as shown by the blue rectangle on the right. Instead, black hole candidates in my sample are identified by the variable visible light signal of their gas. I use data from the Dark Energy Survey camera, which has a 2.2 degree wide field of view. That's more than 4 times the apparent size of the full moon in the sky. The target area of this instrument is more sensitive to spatially extended objects like galaxies. Using variability selected sources ensures that my sample is not biased against off-center intermediate mass black holes. My method could be applied to large survey data sets, like the Large Synoptic Survey Telescope. If a significant fraction of black holes are indeed off-center, it could signify a larger population of wandering black holes hiding in observational data.
Hi, my name is Savana Ammons, and today I'm going to give you a brief introduction into complex analysis, the field of math that I will pursue research in. So imagine for a minute that I asked you to compute the 2 integrals that you see in the middle of the screen. In a way, they look kind of simple. Sure, you might not understand all of the notation involved or even what an integral is, but these problems look so short. They can't possibly be that hard, right? Well, what if I told you that these 2 integrals stumped top physicists for decades, and that it took a complete overhaul of the way we view numbers in order to solve them? Enter complex numbers and complex analysis. Complex numbers are created by adjoining an imaginary number onto the traditional numbers that we're used to, thus augmenting the normal number line in order to reveal an entire plane of complex numbers that were always hidden just beneath the surface. Meanwhile, complex analysis deals with the study of complex functions. That is, those functions that take in complex numbers instead of the normal real numbers. A prime example of this would be the sine function, which you may have encountered in trigonometry. On the top left of your screen, you see the traditional graph of the real sine function. As you can see, it's pretty simple, just a normal wavy line. On the right of the arrow, though, you see the graph of the complex sine function, which is a lot more complicated, as it requires multiple 3-dimensional graphs along with an entire color palette just to be able to try and visualize what this thing is doing. Needless to say, the complex counterpart is a lot more intricate. And for this, you'd be forgiving for assuming that complex analysis just makes problems harder to solve. But that's actually where you'd be wrong. You see, complex analysis allows us to reveal the intricacy that was always hidden within the problem. It's as if before our view of the problem was restricted. but now we get to see it for what it really is. It's because of this that, despite complex analysis sounding very theoretical, it has applications in many hands-on fields such as mechanical, aerospace, and electrical engineering, as well as thermodynamics and optics, the field where the two integrals that I started out with were from. Now, in addition to all of these applications, complex analysis also has some fun applications too, one of the most stunning of which being fractals. So fractals are geometric objects that look basically the same no matter how much you zoom into them. And one of the most famous examples of a fractal would be the Mandelbrot set. So I have two pictures of the Mandelbrot set on the bottom of the screen. The black parts of these images represent complex numbers within the Mandelbrot set, whereas the others are colored according to how far away they are from the Mandelbrot set, according to some measure. I hope that with this talk, I was able to give you a brief understanding of some of the fun and cool applications of complex analysis, and I hope to talk to you in the future about my research in this field. Thank you for your time.
Hi, my name is Yansy Perez, and I'm in the Department of Mathematics. And recently, a new class of inverse semigroup called the inverse semigroup of self-similar graph actions was discovered. And I had the opportunity to characterize some of the important properties of this inverse semigroup. This inverse semigroup is important to a branch of mathematics that in turn have applications in quantum physics. But what is an inverse semigroup? An inverse semigroup is a set together with an operation. So for example, the real numbers together with multiplication, such that each element has what we call an inverse. In the case of the real numbers, 1/2 is the inverse of 2. Our inverse semigroup involves what are called mathematical graphs. And then this next slide, there's an example of two different graphs. A graph is a set of vertices, symbolized by the dots, and set of edges between them, which are the lines. In our case, our edges are directed. A graph is connected if for any two vertices of the graph, there is a path between them. So for example, take B1 and B2. If we take this black edge first, then the top blue and the right blue, then we can get to B2. It is strongly connected if we can also get back. And notice we cannot get back from B2 to D1. However, everything on the red is strongly connected to each other. So we call this a strongly connected component. Similarly, everything on the blue is strongly connected component. Everything on the blue is a strongly connected component. In inverse semigroup theory, it is important to know whether a inverse semigroup is what we call zero simple. And we discovered that if we start off with a connected graph, then our inverse semigroup is zero simple if and only if it is strongly connected. So our graph on the left here is not strongly connected, therefore it is not zero simple. However, it is not necessary for a graph to be connected for it to be zero simple. What we discovered is that if a graph is zero simple, then it has no edges between any strongly connected components. So again, if you look at the graph on the on the left, the blue strongly connected components and the red strongly connected component is connected by the black edges. It's another reason it is not zero simple. However, it turns out that the graph on the left is zero simple, and notice that there is no edge between the two strongly connected components. I thought it was fascinating that we were able to take this new class of inverse semigroup that no one knew much about and were able to characterize some important features. Thank you.
Hi, my name is Margie Ruffin, and I'm a first-year PhD student in the Computer Science Department, and I will talk to you about user perception and image manipulation. Through exploration, we see that media manipulators act quickly to establish their own narratives, whether it is for humor purposes, political purposes, religious views, and the like, by swiftly creating and seeding content to target certain audiences in the hopes of going viral, or even just causing a large enough stir that will alter opinions. The purpose of our research is to create a useful, automated tool that will aid social media developers in combating misinformation in the media, specifically images. Throughout the development process, key questions were raised, including, What are some of the most popular techniques used to alter photographs, and how can we use them to then reverse engineer photos to pinpoint exact locations in them where an alteration has taken place? Also, is there a way to gauge overall sentiment of a photo based on the objects in that said photo? And if so, can we determine if the sentiment is negative or positive that could possibly justify a conclusion that the photo was altered to 1, change sentiment, and 2, for a malicious purpose? As seen in the chart, there's a 4-component breakdown that highlights different manipulation purposes found in lots of images on social media, including meme creation, facial touch-ups, opinion-based, and the section that we would like to highlight, fake news, or a false sense of danger. To be successful, our algorithm performs several tasks to make these classifications. It will scan an image, crawl the internet for a similar image that may or may not have been modified, and compare the two. As seen on the next slide, using computer vision and machine learning techniques that include object detection, text detection, and facial recognition, we can analyze the given pictures for certain features. When one or more of these features intersect with the manipulated regions in the images, we can then determine whether an image should be further analyzed. Following a set of predetermined heuristics and algorithms, our image tool can classify the set of images as modified or unmodified. The modifications are subtle or not subtle, and given the modifications, malicious or not malicious. We believe that this gives social media developers an automated way to perform a task that is otherwise manual. What would naturally take hours to do by searching through droves of internet images, this tool can do in a matter of seconds. To measure the usefulness of the tool, we are also conducting a user survey to gauge image sentiment and to measure its overall effectiveness. Thank you.
Hi, my name is Román Saltar. I'm a first-year graduate student at the University of Illinois in Urbana-Champaign, the Department of Aerospace Engineering. I work with Dr. Villafañe and Dr. Friend on data-driven subgrid scale models for turbulent multi-phase flows. What if I told you that volcanic ash plumes, blood flow, and oil spills all have something in common? They all can be categorized as multi-phase flows. Multi-phase flows are fluid flows that contain more than one phase or component. And understanding these flows can help us predict how oil spills propagate in the ocean, how ash and debris is transported in the air, and it can even help us understand the cause of various heart diseases. When studying multi-phase flows, computer simulations are often preferred over experiments due to their lower cost. However, flows that are geometrically complex and highly turbulent sometimes exceed even the capabilities of modern supercomputers. Mathematical models that predict the behavior of turbulence can be used to simplify and accelerate these simulations, making more complex simulations feasible. My research focuses on a method called large eddy simulations, or LES. On the next slide, you can see a schematic of how LES works. To solve a velocity field, LES uses a filter to separate the scales of turbulence. Once separated, it solves a set of equations for the large scales and models the small scales, as shown in the diagram. I am focusing on developing a new turbulence model using machine learning and deep neural networks to help improve the prediction of turbulent multi-phase flows. Our deep neural network algorithm uses data from previously conducted experiments or simulations to develop models that are faster and more accurate than the available ones. Currently, this method is being tested on simple flows to validate the results. But in the future, it will be tested on flows that are relevant to environmental, biomedical, mechanical, and aerospace engineering, as well as the atmospherical sciences. With the help of machine learning, I hope to revolutionize our understanding of turbulence multi-phase flows.
Good afternoon, everyone. My name is Vanessa Awate. I'm a first-year PhD student in the Department of Aerospace Engineering, and I'd like to talk to you about my research, which I tend to think of as a puzzle, otherwise known as the roto-aerodynamic puzzle. I'm sure most of you have spent some time outdoors with nature within the last few months, and perhaps some of you have taken a closer look at birds in flight or landing. If you have, you probably noticed that a bird performed a fast pitch up maneuver during the perching process to keep it from overshooting the branch or telephone wire. In aerodynamics, that action of rapidly pitching a blade or a wing produces a complex phenomenon known as dynamic stall, which results in a highly unsteady flow field around the wing body. The reason why my research is looking at this dynamic stall phenomenon is because it's prevalent in everyday aerodynamic structures, and it affects the performance of devices such as wind turbines, helicopters that are used for medical transport, etc. As can be seen on the second slide, my primary research area focuses on experimental work. And that experimental work allowed me to perform experiments on the airfoil inside a wind tunnel and be able to characterize the flow within the dynamic stall process by pitching an airfoil really rapidly in the wind tunnel. That action of acquiring data allow us to improve numerical models and to eventually lead to more effective dynamic stall mitigation strategy by controlling the flow around such wings or wind turbine blades. Thanks for your attention.
Hi, my name is Andújar. Have you ever heard of the recent cold weather in Texas and how it led to the failure of their power grid? Maybe you've heard of Hurricane Irma and Maria and their effect on the power grids in islands like Puerto Rico. Our energy infrastructure was designed a long time ago, and our energy needs and the climate landscape look very different. Our energy demands are becoming more complex as electrification and renewables increase and as extreme weather events become more common. It is essential that we modernize the grid to become more resilient and adaptable. A solution is a smart system, a smart city, that is capable of making real-time decisions by monitoring and communicating so that problems can be isolated and solved without affecting the rest of the system. Smart grid is more aware and capable of making control decisions based on the information that's provided to it. One of the challenges to the smart grid is to control bidirectional flow. For example, a building might be producing excess electricity from its solar panels at some period of time, but not enough to sustain itself throughout the entire day. And that's the bidirectionality at work. So we end up with a mismatch between production and consumption. One of the typical strategies is to introduce a buffer, like batteries. When people think of a smart city, they often think about the electric grid, but we can also look at smart heating and cooling networks. Similar patterns of bidirectional flow also happen in heating and cooling, especially when buildings form part of a network within the city district. However, the application of advanced control strategies within smart heating and cooling systems with varied types of thermal storage resources is still in its infancy. My work focuses on trying to design and identify effective control strategies so that the heating and cooling system can meet the desired objectives, like keeping the people inside comfortable, reducing costs, reducing electricity consumption, or just keeping the food or medicine at the desired temperature. One of the aspects that we're interested in is optimizing the location of the thermal storage so that it is located in a way that when it's controlled, it's more effective and responsive. The goal is that using this control knowledge, we can design a better system. Ideally, creating a co-design process where the design of the control strategies inform the design of the actual system, the physical system, and the physical system inform the control strategies, leading to a system that is better prepared for the next heat wave or extreme cold storm. Thank you.
Hi, my name is Anaira Román Santiago, and I am a graduate student at the Chemical and Biomolecular Engineering Department at the University of Illinois at Urbana-Champaign. Today I will be talking about my research, which I titled Electrochemical Technologies for Selective Nutrient Recovery. Now, what does this all mean? So, due to the increase in global population, there has been a need for an intensified crop production process. As many of you know, this is usually done by the use of fertilizers. Now, fertilizers are mainly composed out of ammonium nitrate, which is a chemical compound that has two very important nutrients for plant production. Now, ammonium is typically produced via the Haber-Bosch process, which is a very energy-intensive process that also produces a lot of fossil fuel emissions, like CO2. Now, it's not only a problem of where does ammonium come from, but also where does it go to. If you follow me to the upper right part of this screen, we can further explore this process. So essentially, ammonium is introduced by the use of fertilizers, but only about 60% of the fertilizers are actually uptaken by plants, which means that the other 40% goes to runoff water. Now, although these nutrients are essential for plants, they are actually very toxic to aquatic animals. Not only that, but it's also contaminating our sources of water. And although our water is usually treated prior to our consumption, current chemical technologies aren't able to completely remove these ions from water, which is why they eventually end up in our tap water and we have indirect exposure to it. So what I'm trying to get at is that there's two main problems. We still need fertilizers, but unfortunately, these are also contaminating our sources of water. But what if I told you that we are able to solve both of these problems with just one system? And in this next slide, that is what I am presenting to you. So my research group is looking at electrochemical technologies that will enable us to not only remove these from water, but also recover them. Essentially, we are looking at ammonium selective electrodes that, when applied a negative potential, will absorb these positively charged ions onto the surface and remove them from water. Subsequently, we can reverse the potential, and this will cause the ammonium ions to be further pushed to a concentrated solution that can later be further treated to recover these ions. So essentially, we will have not only the removal of them from water, but we will also recover them and use them as nutrients. Thank you.
Good evening, and welcome to my lightning talk on the dynamics of redox active polymer solutions. My name is Dejuante, or DJ Walker, and I work under Charles Singh in the Department of Chemical and Biomolecular Engineering here at the U of I. All right, so polymers are a type of molecule that consists of repeating subunits. Some common examples are DNA, skin, hair, rubber, and plastic. Redox active polymers are special polymers that are capable of transporting charged species, hence redox active. Redox flow batteries are energy storage devices usually reserved for large-scale applications. So for example, you might use a lithium-ion battery in your car, but you would use a redox flow battery to power your home. Redox active polymers are of huge interest in the field of redox flow battery electrolytes. These polymer electrolytes are expected to transfer charges across the electrolyte interface while it's not actually crossing themselves. In other words, the electrolyte is expected to be both redox active and size excluded. RAP ion transport dynamics within RFB systems are not well understood. This is because these are very complex systems that involve both non-equilibrium dynamics, or other words, flow, and redox chemistry, where the charges are being transferred. So I designed simulations of RAP solutions to serve as a starting point to understanding these more complex systems that will lead to the design of better flow batteries. Thank you for attending my talk.
Hello, my name is Blake Ocampo, and I am a first year in the organic chemistry department. The importance of a molecule's configuration can never be understated, as even point changes within a molecule can drastically alter how it interacts with our body. Nature can take advantage of highly specialized enzymes to generate single configuration molecules. Organic chemists have been attempting to imitate nature's abilities for decades in pursuit of biologically active molecules in a field known as asymmetric catalysis. Over these years of work, catalyst systems have been created that can promote the formation of single molecules over a variety of reactions, unlike enzymes. A high number of these catalyst systems use the molecule I've drawn here, the bisoxazolene, more commonly known as the box ligand. It is a special family of molecules as there are 3 different positions where changes can be made, which can greatly affect the outcome of a reaction. Unfortunately, catalyst design is limited to just chemical intuition. What we need is a method of choosing the catalyst to make, not just a general idea of what things may or may not influence the outcome, and our group has created a method to try and optimize a catalyst through computer science. Our goal is to teach a computer algorithm how to do chemistry, to recognize patterns that we may miss in a way where we don't have to know how a reaction is happening to understand the relationship between the reactant and catalyst. We've discovered a new way to represent chemical space where other methods have failed, and we're able to generate a workflow to predict how a catalyst might behave. This is where my research comes in. The defined workflow on the next slide is as follows. 1, make a library of box molecules that can actually be made. 2, represent how the molecules exist in chemical space. From there, for number 3, select a subset of this library that represent a broad number of different structures with different properties using data-driven methods. And we can call this the Universal Training Set, or UTS for short. After that, for number 4, we choose a system and test how each catalyst promotes the reaction. And number 5, apply machine learning methods to generate models that could predict the best catalyst within the original library. This would lead us to the end goal of finding the best catalyst for the system we chose. I have chosen the copper snap reaction as it generates a very important building block for biologically active molecules. In a survey of compounds pursued by companies like AstraZeneca, GlaxoSmithKline, and Pfizer, the molecules this reaction forms represent nearly a quarter of the structures they were pursuing. If a direct relationship can be generated between observed and predicted outcome, this indicates the workflow works for the optimization of box molecules with copper snap chemistry. It would also validate the model as a predictive tool to allow further expansion to other frontiers of chemistry. Thank you.
Hello, my name is Marya Ornelas. I am a first-year PhD student in the chemistry department, and I'm going to be telling you all about a project I'm working on in the Meta Lab towards the development of a universal coronavirus vaccine platform. So since the creation of the first vaccine in 1798, vaccines have become an incredibly powerful tool in public health, in large part due to improvements in vaccine design that have been aided by improvements in genetic engineering technologies. So in the Meta Lab, we aim to tap into those tools and powerful resources we have available to us to create vaccines that are effective, meaning that they elicit a strong immune response, and also vaccines that are universal, meaning the vaccine can not only be applied to the current pathogen, but also to future variants and even to other viruses or to other organisms. And so to do this, we are co-opting coronavirus biology. So on the left side of the PowerPoint slide, you can see a small schematic of the coronavirus life cycle. So it first recognizes its host cell, and then it injects its RNA genome. And once the RNA is inside the cell, it gets modified in ways to make it more stable, allowing the host cell to read the RNA and make more viral proteins that form the viral particles, which can then exit the cell and infect other cells. And so we aim to make the second step in this pathway, the step of RNA modifications, dependent on an unnatural molecule. And so what this means is that, as you can see on the right side of the slide, in the presence of this unnatural molecule, the virus will be able to propagate as usual, but then once it has used all of that molecule, it will no longer be able to propagate and it would die off. So we aim to make the stability of the virus's genetic code and thus its ability to propagate dependent on this unnatural molecule. And since the step is conserved across coronaviruses, this platform may be able to be used for future variants, as well as in general to other viruses and organisms as well. Thank you for listening.
Hi everyone, my name is Brittany Payan. I am a first-year bioengineering graduate student here at the University of Illinois Urbana-Champaign. Today I will be talking to you all about my project titled A Novel Treatment for Huntington's Disease. Now, Huntington's disease is an incurable and ultimately fatal neurodegenerative disorder that affects 1 in 10,000 individuals. It is caused by a genetic mutation in the Huntington gene that leads to a polyglutamine expansion in the Huntington protein. This overabundance of glutamines actually leads to the formation of linkages between and within each other and leads to the formation of aggregates. Now, these aggregates, through unknown mechanisms, are known to dysregulate the cell's function and lead to eventual cell death. And it's actually the cell death that correlates between the Huntington's symptoms known of cognitive decline, movement disorders, and personality changes. Now, in the past, studies have shown that the removal of this mutant protein can actually improve neuron viability and aid behavior analysis. And so in this next slide, I will talk to you all more about the approach I will be taking on. And so because we know that Huntington's is a monogenic disorder, it is actually a great target for genome editing technologies such as CRISPR-Cas9. And in fact, CRISPR-Cas9 has been used in the past to aid an animal mouse model. And it has actually shown that it can improve the phenotype. But in the end, there's still an eventual death in the model because of a neurodegenerative environment that has already been created by the cell loss population. And so to try and combat this issue, we have turned towards cell replacement therapy, which is using stem cells to recreate this lost population and then inject it back into the brain. And so animal studies have shown that cell replacement therapy can aid in the Huntington's phenotype. But in the end, we still see eventual death. And this is because there are host cells that continue to create this neurodegenerative environment through the mutant Huntington protein. And so my research focuses on using both cell replacement therapy and CRISPR-Cas9 genome editing and put them together to hopefully create a new treatment that in the future can lead to the cure for Huntington's disease. And so with that, I would like to say thank you and have a great day.
Hello, everyone. My name is Catherine Jalomo, and I am a first-year graduate student in chemistry. I will be discussing the use of gold nanoparticles for delivering anti-cancer polymers. This year, the American Cancer Society estimates that there will be 4 people diagnosed with cancer every minute in the US alone. Some good news, however, is that there are new and improving treatment methods, particularly by designing specialized ways of targeting cancer tissues. Tissues are made up of cells, and in order to target cancer cells, we have 2 big questions. First, how are they different from healthy cells? And second, what can we do to use the information to exterminate cancer cells? One differentiating characteristic between cancer and healthy cells is that cancer cells are slightly more acidic than healthy cells. My colleagues and I are working on a way to use this information to exterminate cancer cells through delivery of gold nanoparticles loaded with cargo polymers into cells. This is an example here on the left of a gold nanoparticle, which is made up of gold atoms that are stuck together and coated with molecules that make them more stable and cell-friendly. They also perform a function, which in this case is to target and destroy cancer cells. Imagine the gold nanoparticle is a truck, but instead of carrying its load on the inside, it carries its load on the outside. This truck needs to be a certain size and shape in order to carry anti-cancer cargo into cells. Part of my role in this project is experimenting to find the best size and shape, whether that is long, short, or spherical particles. Next, the nanoparticle truck gets loaded up with large compounds called polymers that my colleague is designing. These polymers are long chains of smaller molecules that we will use to help attack cancer cells and will be loaded onto the outside of our gold nanoparticles. Once we design the gold nanoparticle trucks and load the anti-cancer cargo, hopefully it will go into cells. When cells take in large particles like this, they are often placed in cellular boxes called endosomes. The endosomes from healthy cells here are depicted in blue, and an endosome from cancer cells is in red. My colleague and I are working on 2 ways to get the cargo out of its endosome box. One way is to build an escape plan into the polymer cargo. The polymer reacts with the acid in these cells and falls apart, generating even more acid and causing the endosome and ultimately the cell to fall apart. The second way uses laser light to make the polymer fall apart, allowing it to escape and ultimately kill cancer cells. In the future, we hope to make this applicable for other types of polymers and deliver other disease-fighting cargo into cells. Thank you.
Hi, my name is Howard Weatherspoon, and I'm a first-year PhD student in the Chemical and Biomolecular Engineering department. Under the direction of my research advisor, Professor Baron Peters, I'm researching how to develop population balance models for semiconductor nanocrystal synthesis. Semiconductor nanocrystals are simply tiny light-emitting particles on a nanometer scale, and specifically, we're looking at quantum dots. Quantum dots can be found in the applications of solar cells, display technology, and bioimaging. To understand how quantum dots are synthesized, we look at what's called nucleation and growth theory. Nucleation and growth theory can be seen in the schematic at the bottom of the screen, where we begin with small particles known as monomers, and they're bathed in a solution where they become nuclei. The nuclei then grow, and once they reach a specific radius size called the critical radius, they then transition over to the growth stage. Within the growth stage, nanocrystals can grow and attach themselves to other nanocrystals to become bigger nanocrystals, or they can aggregate back into the solution. And aggregation is just when particles dissolve back into the solution. So I want to keep that point in mind as I move on to the next slide now. We want to be able to build these mathematical models to correlate them to experimental data. And we want to be able to correlate things such as the size distribution, and concentration to the data analysis that comes from like absorption spectra. So these models are population balance equations. And you can think of a population balance equation like keeping track of a population of a country, where I have highlighted in green the accumulation term of the equation. And that's like keeping track of the total population of a specific area. Then in purple, we have a flow in minus a flow out term. And that's like keeping track of the number of people that immigrate to your country or that immigrate from your country. And the last term in red is the generation term. And that keeps track of the birth and death rates of a country. And this is where I brought up the point in the last slide about aggregation or nanocrystals attaching themselves to other nanocrystals. That generation term keeps track of that process within the growth stage. And this developing these models will help us provide kinetic insights to what happens during experiments and how these parameters affect the experiments, such as nucleation temp, ligands, or even reactivity within the experiments. I want to thank you for your time and listening. I hope you have a great day. Thank you.
While diamonds are commonly admired for their beauty, it may not be well known that they're vital to some kinds of scientific research. Hello, my name is Dina Michel, and I'm a first-year PhD student in the physics department. My research will involve using a quantum diamond microscope that I am currently collaborating on setting up by working on its software, and I'll be using it to measure 2D materials and devices that I'll later have the opportunity to construct. Since my project is just getting started, what I'll do today is give a bit of background and discuss what I intend to do in the future. To start, what is a quantum diamond microscope? It's a microscope that relies on the properties of a special kind of diamond that has a very useful defect. Diamond is comprised of carbon atoms, and this particular kind of diamond is said to have a defect because some of the pairs of carbon atoms are replaced by a nitrogen atom and a corresponding empty space that's called a vacancy. And these nitrogen vacancy centers possess properties that make the diamond that has them very useful for sensing intrinsic materials properties and responses to things like applied magnetic and electric fields with much greater sensitivity than many other kinds of measurement techniques. Using this microscope will involve shining green laser light onto the diamond, which we will have sitting on top of the material of interest. When the laser hits the nitrogen vacancy centers, they are excited and emit a fluorescence that we can actually measure. And because the light that's reflected off of these nitrogen vacancy centers will differ depending on what kind of material we are imaging, and because of the nitrogen vacancy center's intrinsic properties, we get really high resolution data about our sample this way. And so what will I be using this microscope to image? There are materials that can be peeled apart or exfoliated, as it's called, into single atom thick layers, and they're called two-dimensional materials because they resemble a sheet or a two-dimensional plane once they're in that state. This class of materials is of great interest to academic research because it's a platform for potentially observing new and exciting physics. It's also of great interest in industry because it can be applied in a wide range of areas from technology to medicine. For example, it can help with improving data storage, batteries, processors like those in computers and cell phones, all the way to eventually helping scientists along the path of realizing quantum computers. So to review, I will be using and constructing in collaboration a quantum diamond microscope, which takes advantage of a particular defect in diamond, nitrogen vacancy centers, to study single atom thick magnetic materials. And who knew that along the way, I would come to learn that diamonds really are a physicist's best friend.
Hello, everyone. My name is Ále Baptista. I'm a first-year physics PhD student at the University of Illinois at Urbana-Champaign in the Department of Physics. And today I'll be talking about distributing quantum information in superconducting circuits. And I'll open the talk with the question, what is a quantum computer? You may have heard some rumblings about Google developing a quantum computer and the United States getting interested in quantum computing as a developing technology. But what do they look like? Well, the answer to that question is what we have on the left here. This is one of Google's quantum computers, their Sycamore chip, their latest quantum computer. And upon first glance, you may ask, well, this doesn't look like that different from a processor that I have in my computer right now. What's the big deal? Well, the main difference between a quantum computer and a classical computer is that a quantum computer uses quantum operations to perform its calculations. And this makes it very optimized for probing quantum questions. Now, quantum physics is the most fundamental physics that we understand. So being able to develop a system that can probe quantum physics very efficiently will help us understand quantum physics with many bodies very efficiently. Now, what's the basis of the quantum computation in our quantum computer? Well, that's the quantum bit, or the qubit. Now, what's so special about the quantum bit? Well, let's revisit the classical bit to understand that. A classical bit is a piece of information. It can be either 0 or 1, and bringing many of these different pieces of information together can form complex pieces of information. In a very similar way, a quantum bit is a piece of information that can be 0, denoted by the red arrow pointing up, or 1, denoted by the blue arrow pointing down. But what's special about a quantum bit is it can also be something in between. It can be this purple arrow which denotes something in between. This is an example of superposition and is the basis of quantum computation. Now, we have our superposition, we have our quantum bit, that's great, but what's the catch? One of the main issues with quantum bits is that they lose information very easily. And this loss of information represents two main challenges in quantum information today: the difficulty of scaling quantum computers with quantum bits and the difficulty of sharing information between quantum computers. And that gets into the niche of my research. So in my research, we ask, how do we connect qubits without losing the precious information that's stored within them? And in our lab, we're taking a bottom-up engineering approach to understand how we can design connections between qubits that don't lose the precious information that we have. Now, we're just getting started, but we hope that through this research, we'll be able to create easily scalable quantum computers. And in understanding the connections between qubits, we might be able to understand how to connect different quantum computers together better as well. And connecting different quantum computers together would create a quantum network of information, which would represent a very powerful tool for tackling some of the most fundamental physics that we know today, quantum physics. Thank you very much.